I will start by making the case for life being too complex for one scientific discipline, and that we have to approach this problem from many different aspects. Today, it is popular knowledge that an egg cell and a sperm cell combine to give rise to a human being. But this took us a while to get there. At the end of the 17th century, for instance, preformation was the prevailing theory of embryogenesis. You pretty much start out in your final shape in the egg or the sperm cell depicted here on the left and just grow. It took many different scientific disciplines in the 20th century to answer the most fundamental question. How do the living organisms reproduce? Today, it is textbook knowledge. Living organisms reproduce themselves by transmitting genetic information to their progeny. When I show a typical schematics of a eukaryotic cell and ask, where is this genetic information? I'm assuming your eyes focused on this friendly-looking fella in the nucleus. When I ask what is in there, you are most likely thinking about DNA or chromosomes, a picture also known from popular media. And when I say living organisms reproduce themselves and show a movie like this, you know you are watching cells dividing, even though you may not know all the details in this particular movie. The information that DNA seen here in gold is the hereditary material that is duplicated and divided up between two daughter cells is an information that we take for granted in the 21st century. Well, even though we humans are on this planet for about 200,000 years, we have figured out this fact quite recently. So let's go back to our typical cell and ask, where is the hereditary material? Erwin Schrödinger has postulated in his famous book, What is Life?, that a gene is probably a large protein molecule in which every atom, every radical, etc. plays an individual role. Why did people bet on proteins and not on DNA? Well, people already knew that DNA is a simple polymer when compared to proteins that are far more complex in their molecular composition. Let's take a quick history tour to see how scientists from many different disciplines put together the pieces of this amazing puzzle. I take you back to 1918. The world has been ravaged by the World War I and the Spanish flu. The flu eventually killed more people than the act of war itself. Majority of fatalities during the pandemic were not caused by the virus acting alone, but the pneumonia that developed as a consequence of the viral infection. This sounds very familiar to what we are hearing about the current corona pandemic. As it is the case today with an army of scientists trying to develop a vaccine against the coronavirus, Frederick Griffith, who was a bacteriologist and a physician, worked on a vaccine against pneumonia. With the benefit of hindsight, we can now say that Griffith made a very influential experiment with two strains of bacteria. One is infectious, disease-causing, and the other one non-infectious and harmless. When he injected the mouse with the non-infectious rough strain of Streptococcus, nothing happened. Mouse was alive. It's called the rough strain simply because the surface of the bacteria looked rough. When he used the infectious bacteria with the smooth, shiny appearance, the mouse died. So far, no surprises. Also not a surprise, when you heat the infectious smooth strain and kill the bacteria, the mouse is happy. Now comes the killer experiment, literally and figuratively. He mixed the heat-destroyed infectious bacteria with the non-infectious bacteria. The mouse died, even though the infectious bacteria was dead. Not only that, Griffith found living smooth bacteria in the blood of the dead mouse. Well, how did the heat-destroyed infectious bacteria resurrect all of a sudden? Griffith concluded that the rough strain bacteria has taken over the transforming principle from the heat-killed smooth bacteria and became virulent. Well, what does this mean? Fast forward to 1944. A physician A biochemist and a geneticist set out to identify the transforming principle that was observed in Griffith experiments back in 1928. Avery and colleagues simply asked, what part of the bacterium is infectious? They heat-killed the infectious smooth bacteria, represented here in red, and purified out the major components, proteins, lipids, etc. Then they mixed the purified components with the non-infectious rough bacteria, represented here in green. As you probably already guessed, only the DNA fraction transformed the harmless green into the infectious red. They did clever experiments to further support their conclusions. For instance, they used substances that would specifically destroy the RNA or the proteins, etc. The green would still turn into the red color, or the non-infectious cell would still become infectious. But when they specifically destroyed the DNA, the transforming activity was abolished. The problem with assays like these is that you can never 100% exclude the possibility of co-purified impurities that cause these effects. What if trace amount of something else, maybe a protein, tags along the DNA and is responsible for the observed transforming activity? So this brings us to our last station before the happy ending. In 1952, Hershey and Chase, a bacteriologist and a geneticist, came up with a different idea to test this model. What they did is they have overly simplified the components of the experiment. It was very well known back then that a type of virus called bacteriophage is the natural enemy of bacteria. Here you see a bacterium being invaded by dozens of phages. The point here is that the composition of a bacteriophage is exceedingly simple. This matchstick man, if you will, here in the top right corner, carries its DNA in its hat. The rest is a protein code. That's it. Hershey and Chase knew that something gets injected into the bacterium that carries the necessary instructions or the transforming activity to make more of the virus inside the bacterium. Well, what is it? Protein or the DNA? So they made use of radioactive labeling, and they specifically labeled the DNA and the protein. At the end, they have concluded that it must be DNA, which is a basic textbook knowledge today. Depending on the type of virus, it's either DNA or RNA. In their case, it was DNA. A particularly infamous example of an RNA virus is the coronavirus that we are desperately trying to defeat today. But more on this later. Well, at the end of the day, you can still keep making the same arguments of trace amount of impurities tagging along and being responsible, and so on and so forth. But these are now too many indications to ignore. You probably did not know much about the work up to now, but the next one, you know. A truly interdisciplinary effort by chemists, physicists, and molecular biologists put the final nail in the coffin. In 1953, Watson and Crick published their one-page landmark paper in Nature, in which they correctly predicted the structure of DNA, the world-famous double helix. When you look at the title itself, it sounds quite underwhelming. Molecular structure of nucleic acids. It was already clear that the DNA was an unbranched polymer. So now we have a prediction for its structure. So at the end, we have a sugar phosphate backbone, and we have base pairing with 4 different bases. So not that many to really be responsible for rich hereditary information. But the more important sentence is found at the end of the paper. It has not escaped our notice that the specific base pairing immediately suggests a possible copying mechanism for the genetic material. They got it. So join me in the next lecture where we try to understand what it is that didn't escape Watson and Crick's notice. At the end, I would like to leave you with this very famous saying, seeing is believing. Well, mitotic chromosomes were seen under the microscope several decades ago, and today we learned that seeing is believing, not understanding.